Welcome back to Lecture 2 in the series on cell division. And we just talked about how bacteria can reproduce through binary fission. They just divide. And in eukaryotes, in the end, they just divide as well. So the process of cell division, more accurately, nuclear division, is called mitosis. If you recollect, we looked at the cell cycle and we said that there are 3 or 5 phases of the cell cycle. G1, S, G2, all making up interphase. Then we get to mitosis and then finally cytokinesis. Well, mitosis has 4 phases: prophase, metaphase, anaphase, telophase. And back in the day, we used to simply teach mitosis as cell division. All right, and we would say, remember IPMET, interphase, prophase, metaphase, anaphase, telophase, and then in telophase, we said that the cells divide. So we've made it a little more complicated, but in short, it's not so complicated. So I've got a cool series of videotapes for you guys to watch. There's lots of diagrams for you to do, but let's talk about those important features of the of mitosis. All right. So during interphase, remember the cell could be in any one of interphase, and I wouldn't be able to tell. Could be in G1, could be in S, could be in G2, but I can't really see anything. It's just a normal looking cell. In interphase, I just see a cell. The difference between interphase and prophase is that for the first time, I'll be able to see chromosomes. All right, so the first feature of prophase is for the first time I see these dark bodies, chromosomes. If you recollect back from our lectures on the cell structure, there's some structure called the centrosomes. And in prophase 1, the centrosomes start to migrate. They're normally next to the nucleus. They're bundles of microtubules, and they're going to end up forming the structure called the mitotic spindle. In fact, as they move apart, they're really sort of growing these microfilaments and microtubules, which is kind of fascinating. Anyway, so prophase. Centrosomes begin to migrate, building the mitotic spindle. Chromosomes appear for the first time, and then once those chromosomes appear and they're done forming, that nuclear membrane begins to break down, okay? And it exposes those chromosomes into the cell cytoplasm. During metaphase, the chromosomes line up in the middle. Meta means middle. The mitotic spindle's fully formed. It's made up of microtubules and microfilaments, and at the poles of each end, the centrosomes have now be called centrioles, and they form the mitotic spindle. They're anchors to the mitotic spindle, and that's it. It's sort of like the hallmark of mitosis when you look at slides of it. In anaphase, those chromosomes are pulled apart by the mitotic spindle at their centromeres. There's special fibers called kinetochore fibers that are connected specifically to the centromeres. And the chromosomes can sort of be seen dragged through the cytoplasm. Telophase, things start to go back to normal. The chromosomes are pulled to the opposite poles of the cell. Nuclear envelope begins to reappear. Chromosomes begin to unwind. Mitotic spindle dissolves. And then you can have the formation of this structure called the cleavage furrow, which is going to be the place where the cell finally breaks apart. It breaks apart during cytokinesis, and that's it. Now, if you watch these videos, you'll have a better and better idea. And the more you do your diagrams, the more familiar you'll be with the process. So again, in animals, we see the same structures that we see in the plant cells with a couple differences. Here we go. Prophase, I start to see these accretions called chromosomes, dark bodies. The nuclear envelope disappears. In metaphase, and sometimes we break it into prometaphase, but you can see the chromosomes. In metaphase, the chromosomes are lined up in the center of the cell. In anaphase, they've been torn apart and they're moving towards the poles. In telophase, things are going back to normal. Nuclear envelopes beginning to reappear. Those chromosomes will unwind. Now, in the case of plant cells, they don't have a cleavage furrow. They form something called a metaphase plate. All right, so there's two real differences between mitosis in plants and mitosis in animals. The formation of a metaphase plate during telophase, and then these guys don't have those centrosomes. So those are two differences between plant and animal cell mitosis. Here's animal cell mitosis again. Interphase, just a cell. Centrosomes are next to the nucleus. Prophase, chromosomes appear, nuclear envelope dissolves. Mitotic spindle begins to form, driving these two centrosomes to the poles of the cell. In metaphase, they're all lined up in the center, the chromosomes are. There are fibers, special fibers attached to the centromeres of the chromosomes, called kinetochore fibers, and they're going to be the ones that pull it apart in anaphase. In anaphase, they're pulled apart. In telophase, things go back to normal. Nuclear envelope reappears, mitotic spindle breaks apart, cleavage furrow forms, and those chromosomes will begin to unwind. And then finally, the cell divides in cytokinesis. Again, cytokinesis in animal cells, cleavage furrow, cytokinesis in plant cells, we have this cell plate formation. Well, that's mitosis in some detail. And the sad thing is we're going to teach mitosis with meiosis. Now, mitosis is happening all the time, all over your body, right? Cells are constantly undergoing division. But meiosis is another process. It's similar to mitosis because cells divide, but it's really only happening in one place in your body, your gonads. That's right. Meiosis is the process which produces sex cells. So your gonads are your reproductive organs. Your primary reproductive organs, believe it or not, is not your penis or your vagina. It's your testicles and your ovaries, and that's where meiosis happens. So, some big differences between mitosis and meiosis is that in mitosis, or somatic cell division, I have a diploid cell here, a 2N cell that makes two 2N cells here. So I have 46 chromosomes that makes two cells with 46 chromosomes. I know the numbers don't add up there, but that's how we talk about it. In meiosis, I have a 2N cell that makes two 2N cells that finally divide into four 1N cells. All right, so in a sense, they're very similar. Meiosis is a lot like mitosis in that it's just two replicative cycles, right? I go from one, I split to two, then I take these two and split to four, but there's some important differences. Remember, these cells are only haploid cells, they're 1N cells, they're gametes, they're sex cells, they're sperm, they're egg, they're pollen, they're ova, etc. So remember that little n represents the number of pairs of chromosomes for any organism. So remember that n for humans is 23. N for shrimp, 46. Diploid cells are 2N. Therefore, we have 46 chromosomes. Haploid cells are 1N. Remember that a zygote is a fertilized egg. All right, when a sperm meets an egg, then I have a zygote. Gametes are those haploid sex cells, sperm, egg. Horse taco. It's a taco for a horse. Fertilization is the union of egg and sperm. Sexual reproduction. Why is sex so great? Well, it has to do with variability in the cells, and we'll talk about that in a second. So meiosis is a process in which we get from diploid cells to haploid cells. It's got a lot in common with mitosis. In fact, if you know mitosis, you sort of know meiosis, and that's because meiosis is simply two replicative cycles of mitosis, sort of. I could tell if I'm looking at meiosis because it has names like prophase 1 or metaphase 1 or prophase 2, metaphase 2. In fact, it's fairly straightforward. We have prophase 1. Guess what happens? Chromosomes condense, nuclear envelope dissolves, and those centromeres or centrosomes start to migrate. There is one really important thing that happens from prophase 1. We'll get to in a minute. Metaphase 1, chromosomes will line up at the center. In anaphase 1, they divide. In telophase 1, they separate. And then that happens again. Prophase 1, the chromosomes, or metaphase 1, the chromosomes line up. Anaphase, telophase 2, they break apart until finally end up with four new cells. And of course, those cells are going to be gametes, those sperm or those eggs, and you can see those things up top there. Here's another way to look at it, right? I go from one cell to two cells, 2 cells to 4 cells. We divide it into meiosis 1 and meiosis 2. If you know mitosis, you sort of know what meiosis happens. There's some major differences. The first major difference has to do with what goes on during prophase 1 of meiosis. Remember that back in mitosis, the homologous chromosomes, that is the chromosome number 1 your mom gave you and the chromosome number 1 your dad gave you, line up along the metaphase plate in metaphase. Well, in prophase 1, those homologous chromosomes, that chromosome number 1 your dad gave you and its sister chromatid and the one that your mom gave you, the 4 of them get together to form a tetrad. And in the tetrad, because these guys are so similar, I can have pieces of mom's chromosome number 1 and dad's chromosome number 1 undergo an exchange called crossing over or recombination. So in prophase 1 of meiosis, homologous pairs of chromosomes undergo synapsis. They stick together. They form a tetrad, and they can exchange DNA in a process called recombination or crossing over. That's really important. And that leads to an important aspect of the variability of those gametes. I'll show you in the next slide. Prophase 1, I've got tetrad formation crossing over on. Metaphase 1, those guys get up on the metaphase plate. In anaphase 1, they're torn apart, and then telophase 1, they separate. Here's some clear illustrations. I've got this exchange of materials here. I have exchange of materials here. There are 2 homologous chromosomes. This is chromosome number 1 dad gave you with its sister chromatid. Chromosome number 1 mom gave you. Since they're so alike, they can get together on synapsis and exchange materials. And that has some important outcomes when I finally make sex cells. The truth is, the sperm that I make only has 1 copy of chromosome number 1. And it isn't the chromosome 1 or a copy of the chromosome my dad gave me or my mom gave me. It's a combination of the 2. That's why sex is so great. There's so much variability. Meiosis 1 again. Remember, prophase 1 of meiosis, recombination, synapsis, tetrad formation. I see chromosomes. They form a mitotic spindle. They line up. They're torn apart. In meiosis 2, similarly, the process just happens again. Chromosomes line up. Metaphase 2, they're pulled apart. And then finally, I have the cytokinesis where I form those gametes. Now, there's a couple differences between how gametes form in males and how gametes form in females. Remember that in both cases, I have 1 cell that forms 2 cells that forms 4 cells ultimately. Well, that's not totally true in females. In fact, men make lots and lots of sperm. So I have this original sperm cell that's in your testicles called a spermatocyte. It gets turned into sperm. And each spermatocyte cell splits into 2 and then splits into 4, makes 4 sperm. In fact, boys make 2 to 3 million sperm a day. Girls, how many eggs a day do they make? Well, they don't make any a day. In fact, it takes 28 days for girls to make not 4 eggs, but just 1 egg. In fact, oocytes are the progenitor cells in females in the ovaries. You might have heard that you're born with all the eggs you have. That's not really true. You're born with all the oocytes you'll have. And each month, because of the fluctuations of the hormone estrogen and progesterone, you turn one of those oocytes into 1 mature ovum. That oocyte does split from 1 cell into 2 cells and 2 cells into 4 cells, but it only makes 1 viable ovum, and then the other 3 cells form things called polar bodies. You can learn about that in anatomy and physiology. Well, that's it for mitosis and meiosis. Remember that the key importance of meiosis is it increases genetic variability in the offspring. And if you're interested in the topic, take anatomy and physiology with me next year, or with Mr. Del Prado or Mr. Jacobs. Thanks. Oh yeah, boom, that's it.